a faculty member, assistant professor at Tehran University of Medical Sciences. So my PhD was human anatomy, and after that, I was during my postdoc at Yale School of Medicine, I was working on regenerative medicine with stem cell therapy, with adipose-derived mesenchymal stem cell in human and animal uterus. So after that, when I came back from US to Iran, I continued my work, and I worked with stem cells and different type of hydrogel to regenerate the uterine and also cervix tissue. So I had the honor to participate to one of the Leniza, I think. Yeah. Yes. Lecture. It was so good. That's why, and I actually learned a lot from her. So I've decided to invite her, and she was generously accept our invitation. And she, right now, we are together. We will start. Just I want to give a brief introduction about my dear Leniza, about her background. She has graduated from during the bachelor from biology in 2008, and after that she has specialized in molecular biology and genetic in health science. Afterwards, she started her PhD in the postgraduate program in the Department of Morphology and Genetic from Federal University of Sao Paulo. And right now she's a successful lady because she has her own company, which name is BioCytocorn, and she's working as a consulting for cytogenetic and genetic in her company. And she has a very good knowledge and experience in the genetic, with emphasis on the biotechnology, cell culture, and human genetic, and medical and oncohematology. She's working on the different fields, which mainly is genetic and stem cell culture and cryotyping and bone marrow transplantation, peripheral blood stem cell, and infertility, which is our main interest, that they're working on pregnancy loss and recurrent implantation failure and different techniques. So it is my pleasure to have Leniza in our meeting, and I want you please to start your lecture.
Okay. So I'm very thankful, dear Masume, for I've been here now, for the invite, and this our world of the stem cells is very interesting. Is very for our being scientists is very how can I say, fascinating. It's a fascinating area to study, to observe, and they had a lot to show for us. So I've been studying for some years this tissue, and every time I learn something different. So today I will show you the most beautiful studies that we are doing in UniFAS, the university that I'm doing my PhD graduate students. And I want to thank all the people involved in this too. So the class is streamlining stem cell extraction. I've been studying this for about 5 years now, and finishing my PhD graduating study now. And I will show you about, let's only correct something here. Okay. Now you can go. So I'm now available for partnerships, conversations, and whatever else is possible in these fascinating areas. How can I say? I love these studies. I think the cells have much to show us, to many things we can learn about them in ways that the science doesn't shows and believes in all these things about the cells. I think now today cells are a technology in our bodies. Okay, so what's the problem? As we know, the possibility of these cells, I had worked with adipose-derived stem cells. They could acquire genetic abnormalities, compromising their functional capacity in the host tissue within advanced therapies. So, thinking broadly about the flow of all manipulation of cells intended for cell therapies, and considering that these cells will remain in intense proliferative activity, and well knowing the cell cycle while taking into account the biosafety that these cells must present at the end of all process, the entire isolation process must be very well designed. So we know that prolonged in vitro culture of stem cells can result in chromosomal abnormalities because the cells researches for adaptation, and these abnormalities could bring them a selective advantage, as we know, and nature process of the biology, of the cell biology. So here we present, and I want to show you an improved methodology for isolation of this kind of cells. And all these steps presented in were selected and tested in real case, okay? And each part of the protocol process was chosen with cell viability in mind. And when we think about the viability of the cells, we think in the first time in the plasmatic membrane. The plasmatic membrane is essential being in a healthy way. And all these steps is to maintain the health of the plasmatic membrane, okay? So, all the time. As we know, in the literature shows this, human mesenchymal stem cells are increasingly under consideration as they are useful in both basic and applied research. These cells are adult cell type that overpasses ethical issues. That's because they are so interesting to study. And autologous tissue regeneration, engineering, and cell therapy in areas like neoplastic, degenerative disease, and therapeutic applications. So as in the reconstructive surgery, they are very interesting too, because they are an abundant source to obtain viable cells, okay? This abundant source of mesenchymal multipotent and pluripotent stem cells are in the stromal-vascular fraction, as we call SVF, in all the adipose tissue, really. So, as we know, they are multipotent capacity of differentiation, they are polyimmunogenic, they have a high ability of integration in the host tissue, they have a high ability of interaction with the surrounding tissue, and the most important, the immunomodulatory effects too. So, talking more about the methodology, here we had trained a medical team to perform a specific kind of liposuction of the adipose tissue. This team well trained by us, by our team in the university, they made the liposuction performed in a manual way, because in the literature we see the manual way is the one that most preserve the plasmatic membrane too. Okay, so adipose tissue processing methods during and after the surgery can directly affect the cell viability, and so the final cellular yield, the montant, the volume, the final volume of the cells that we can get. And for obtaining cells populations with high viability, literature shows that mechanical treatments, including ultrasound, should be avoided because they can break down the adipose tissue, they can break down the plasmatic membrane. So the manual fat aspiration method with syringes in each turn has been shown to be less deleterious, causing less cell destruction. The difference between techniques can lead to as much as 70% decrease in the number of viable ADS cells. And it has been shown that the tumescent liposuction, tumescent is the one that we use saline solution with adrenaline to prevent bleeding, okay? And this combined manual liposuction and use of tumescent saline with adrenaline yields a great number of cells and with the best quality. Here I will show a part of it was a video we did. I think I think it's charging all the time. No. So here you can see, as you can see, the medical team and I was present in all the steps and all the time in the surgery, observing the steps, observing all the time all the mechanical force, all the manual manipulation, to prevent all the things that could be, how can I say, break the tissue tumor, okay? So here is the protocol. I will, I really made a resume. If anyone has the interest, more interesting, can be send me an email that I could provide the whole protocol, okay? So first, after the surgery, the liposuction, the manual liposuction, the bag was left resting for 5 minutes inside the laminar flow chamber for precipitation of the Grazer layers. We call the bubbles of the adipose tissue, okay? And separation of the tissue containing the cells of interest. Then a sequence of tissue washes was performed. During the first wash, 100 milliliters of Dubelx phosphate buffered saline with calcium was injected into the bag and handshaking with repeated movements, slow movements from one side to another. It then let stand for more 5 minutes. In this time, most of the basal liquid that precipitated was removed and discarded with a 60 milliliter syringe attached into the bag adapter. During the second wash, 60 milliliter of the DPBS with calcium now was injected into the transfer bag and shaking movement was repeated. And then it was left resting for 5 minutes. And all the basal liquid was withdrawn. This process was repeated twice. 100 milliliter of digestion solutions were added to the bag. This digestion solution was made from 93 ml of calcium-free DPBS plus 60 microliters of calcium chloride plus 7 ml of 0.075% of collagenase, okay? And left at 37 degrees Celsius for 30 minutes under slow stirring. All the content of the bag was transferred to 5 ml Falcon tubes and centrifuged for 10 minutes at 400 xg. So, the supernatant was removed and discarded. And I used to add now the Dubelx modified Eagles medium, demand medium, supplemented with 20% of fetal bovine cereals, FBS, as we know, so added to this cell pellet. Viable cells were counted using the triple blue dye exclusion assay and a fresh prepared solution of 10 microliters triple blue at 0.05% in distillated water was mixed with 10 microliters of cellular suspension for 5 minutes. And then the viable cells were observed in microscope and counted in the Neubauer chamber using in here the inverted light microscope. So this cell pellet was equilibrated in cryoprotective medium with FBS plus 10% of dimethyl sulfoxide at a concentration of 1 million cells per ml, okay? So cells were placed in cryo vials and cooled using a freezing container. We using here the Mr. Frost with a cooling rate of 1 degree Celsius per minute to minus 80 degrees Celsius. And then the samples were stored in standard cassette boxes and immersed in liquid nitrogen vapor phase at minus 165 degrees Celsius. Okay. And with all the study readings and knowledge and observation about cell cultures and knowing the cell cycle in mind, the highlight of every technique is the preservation of the plasmatic membrane, as I had already said. And it was because why this plasmatic membrane is so important in here, and all work that we could make with live cells. It's the membrane, the plasmatic membrane, that initiates all the signaling for the nucleus to work, the DNA and all around. It's the plasmatic membrane that has the primary control of the pathways, okay? This we have always to be in mind when we are working with live cells, okay? And the most important thing in the technique is the gentle manipulation. Gentle manipulation all the time that we are working with live cells. Oh, sorry. Here. Little water. Since we are working with live cells, the manipulation is essential. I have seen many laboratories where technicians handle culture bottles inappropriately, closing the incubator door tightly, generating unnecessary knocks, shaking too fast the flasks with live cells inside. Some of them are not in mind that all the process is a mix of steps that are important to maintain the viability of the cells, to maintain the healthy membrane plasmatic of the cells. Okay, so in my work in biocytochrome, I had received some groups that, oh, help us! Our cells are not functioning. Our cells had too many genetic abnormalities. So when I went there and observed all the steps, they are very working with the flasks, working with the protocols of in all the flow of the process, very fastly, very nervous sometimes. And these are very important when you are working with live cells. Okay, since we corrected these things in technique and technicians, the cells initiated to respond better in viability and not acquiring genetic abnormalities. Okay, I used to call this part of the process the manipulation and the a good training of the team, the 4D factor. Since we have 3D cultures of the cells nowadays, and they are very important for us to understand how an organ works, how the cells team works. All these things about manipulating is important to observe, okay? So here I show you our cultures in letter A. When the cells started to get the adhesion in the flasks, okay? And so this part is important to preserve all the viable, the best cells that we had isolated. They started to function in here. This primary culture is very important to maintain this good manipulation, okay? And in letter B, our counting in new biochamber, showing the very good yield that we had after all good manipulation of the cells since the liposuction in the surgery, okay? And some conclusions. These methods demonstrated a high efficiency and less time demanded for obtaining the stromal vascular fraction, and leads a total of 66 minutes with an isolation yield of average of 15.7 million 700,000 million cells per ml, okay? Inside the literature, it's a very good yield as when we compare. And according to the literature, isolation of this kind of cell can take about 3 to 8 hours. So 66 minutes is a very good time for us that sometimes has to emergence to deliver these cells to patients too. Thus, the gain of time allied to a high cellular income is critical for regenerative medicine therapeutics advancement, as we know, and this modified procedure for obtaining higher yields of viable cells was more efficient regarding timing cellular yields than the techniques from previous studies. And even after a long period of cryopreservation, viable cells in this kind of cells, the adipose tissue, were found in the SVF. We had maintained 2 years of cryopreservation, and even after this time, the cells were viable and returned to proliferate in cultures, in long cultures, long time of cultures. So it's a very good idea. Think about this 4D factor, since DNA sequence respond to cellular perception stimuli in the medium, in the medium of they are maintained, the change of the medium, and all these movements inside the laboratory with the team, the staff of the laboratory is very important. Science had demonstrated already that the DNA responded to that kind of stimuli, the perception of the ambient, okay? So I want to thank a lot my chief, Maria Isabel University, my my professor of my heart, Andrea, that in here too, that helped a lot in my beginning of these studies, and Ms. Masume for the invited, Tiran University for the invited and for the opportunity too, and the patients that donate this biological material for this research. And I want to thank you, my family and friends, who supporting me for being here today too. Okay, thanks. And if anyone has
Thank you so much.
questions, thank you.
Thank you, Lenisa, for your great lecture. Thank you so much. And I want to say hi to your mentor and the Professor Andrea, which I didn't know him. Thank you for your attendance here. And thank you for joining to this lecture. It was a very great job that Lenisa and you have done. And we learned. I just have a very few questions. If anyone has question, please go ahead and ask. Otherwise, I have some question I just want to ask from Lenisa. Does anyone has any question? So I'm wondering about, you have discussed about it took like 66 minutes for you to prepare the SVF, right? Through this process.
Yes.
Okay. And you said in the literature, it took 3 to 6 hours to be prepared. Are you considering the culture like the adipose harvest from patient and mechanical digestion, and after that bringing to the lab, or only in the lab you have 66 minutes?
Only in the lab. We took 66 minutes. In the surgery, how we depends on the medical team and all the, how can I say, the documents to enter and to go inside the hospital. Sometimes leads 1 hour, 2 hours, but in the surgery, about 30 minutes to get the 100 ml of adipose tissue, the liposuction, and after in the laboratory 66 minutes to get the SVF fraction. Okay.
Okay, so and are this just, I want to know a little bit about the Brazilian rule. Like, are you using this SVF after that for human treatment, or you are just doing some experiment on it?
Here we can use SVF, but so had, how can I say, little here in Brazil, the protocols are only had the permission of being this if the patients does not to have any kind of treatment that serves him. Okay. Sorry for my English. I was not so trained today.
No, you are very good.
So few places, few hospitals had disinvolved this kind of work because of this, because the treatments are the conventional treatments, and so when the patient doesn't respond to these conventional treatments, they are free to choose the cell therapy.
So you mean that the FDA in Brazil gave the permission for the doctors and physicians to use this type of treatment for human, right?
Yes, yes. But
All
when
right.
we use the SVF only, does not to necessary to do the karyotype. When I got the SVF, I put in the patients without the first time of culture. Okay?
Right.
And when I
You mean
need
the fresh
to
SPF?
fresh SPF, yes.
Right.
When depending on the protocol, on the disease, on the state of the patients, yeah, and I have to do more cultures, a long time of culture, 2 months, for example, 2 months of culture, this kind of cells, I had to do the karyotype only to check
Right.
the biosafety, and then I can transfer these cells to the patients.
So it seems that you have also the SVF banking for patient for further usage, like in the future, right?
Yes, yes. Not so much banks in here. In Brazil, I feel that I start the model to work, but some because of the high costs to maintain the laboratory, they they the industry does not have the interest to continuous to develop this kind of work. And then in
Right.
the university, some universities in Brazil are being not receiving the all the costs to maintain this laboratory. It's very I've been very sad with this because we had lots of good scientists working with this kind of cells, and now they are traveling to another countries to continues their works because of these two.
Yeah, it's happening same here also. Like here, the lack of budget to continue the project, so people are leaving and going to do the rest of project mainly in United States of America.
Okay, okay. It's so sad for our sciences, our country's sciences, because
Yeah.
they're very good science. But
So, I know that SPF contains several population of stem cells, like progenitor cell, hematopoietic stem cell, and adipose-derived mesenchymal stem cell, and mature adipocyte cell. So while you're just digesting mechanically and also with enzyme digestion, the whole population will be inside the SVF. Are you like doing any sort of cell sorting to just use only mesenchymal stem cell, or you are using whole SVF for the patients?
I used to think that the first time of culture is sufficient to separate, and I prefer to use this biological technology. Né?
Right.
Yes. How can I say? Not to put more stress in the cells, passing for many equipments or passing them in many more steps in a protocol. I prefer to finish this separation, and I only get the stem cells really in the final of the first primary culture. And in my observations, I think they maintain for more time than in cultures viable cells and good plasmatic membrane morphology than if I continue doing some steps that some laboratories makes, and I think this causing in the final good results. Do you understand?
Right. Yeah, yeah,
Okay.
that makes sense. I understand. Okay, thank you so
Okay.
much. Thank you for accepting your invitation. It was very good, and I'm hoping we could do further collaboration in future in different
Oh.
fields like stem cell, exosome therapy. Here, right now, we have started exosome therapy, and we got very exciting results in uterine regeneration and also cervix.
Oh.
Maybe at some point I can also give lecture about that. Uh, and yeah, I also used adipose-derived mesenchymal stem cell. It was very good. So this your great lecture remind me all the thing that I have done previously, and it has a great value. Like teach me how to continue. Thank you for your great.
Okay, good, very good. I'm very happy to this and to collaborate for all these kind of studies. And do you karyotype the cells too in any protocol? No?
No, because we are using the fresh cell. That's why we are not doing any
Okay,
karyotyping. Yeah,
okay.
but maybe we can think about it for future. If we culturing the cell, we have to do because we have to preserve the genetic profile of cells.
Okay, it's interesting. Okay, very good.
Anyone has any question? I don't think that. Does anyone has any question? We would be happy to and to have your question. Otherwise, we want to say bye. It seems that
Okay,
no one has a question.
okay. Thanks.
Okay, thank you so much.
Someone.
Oh, all right. Thank you, Surush.
Arian want to.
Hello.
Hi.
Hello.
Hi. Good evening, Dr. Mahsouba and Dr. Leniza. Sorry for interrupting you, and thank you for both of you for your fruitful presentation and lecture about the stem cells. Actually, my question is not about adipose derived stem cells. I wonder if I could ask something about keratinocyte stem cells. If
Sorry,
you
what?
have a keratinocyte stem cell.
A keratinocyte, right? A skin stem cell.
Yeah. If you can answer me or if you could help me in this issue. I'm a PhD student in Tabriz University, and I would like to isolate keratinocyte stem cells, but I have a problem with medium. I cannot find keratinocyte-free FBS medium. I have to use DMEM medium with bovine serum albumin. And I think you cannot get a good result with DMM. If you have information about keratinocyte cell culture, just giving me some advice.
Okay. And I think this kind of cell, do you isolate it? How do you isolate with the insert in Petri flask?
In
Do you
flask.
put it? Okay, because I think depending the volume you put the first in culture, the cells can stress more, how can I say, faster than we wait. Okay, sometimes you had to how can I say, to put minus supplemented medium in beginning, because they are more the tissue maintain to the vitamins, the proteins they need, and then when are more how can I say, spreading the flask, you put more supplemented medium. I think if you put it all together, the tissue to proliferate the cells, and too much medium supplemented the FBS or lysate, it can accelerate the cell cycle too much. So wait for the flask to be more these cells more separated from the host, the first tissue, and then while you are cleaning the cells that are not the stem cells, you put the medium supplemented much more percentage, high percentage in how can I say,
FBS.
The FBS or the lysate, to not to shock, not to stress the cells. Okay, did you understand, Arian?
Yeah, Dr. Leniza, thank you so much.
Okay, if you want to send me an email explaining better too, I think I can help you better too, because you can show me how do you isolate the cells. This step of the protocol is too much important to decide and the mediums use the reagents to decide this second part of the protocols. Okay, if you want to, you can send me an email explaining better.
Thank you so much, Dr. Leniza. Thanks for your help.
Thank you.
I think, Arian, what I can also add is that Linza means that you have to put your cells in a few medium at first, and after that, when cells just settle down in the flask, then you can add more media. It's like if you put whole media together with high percentage of FBS, the cell will not just grow properly. They should have a little bit starvation, and after starvation, they will get the second media more deeply, and they will start to grow. And I'm sure through the email you can get more detail from Leniza.
Yes,
thank you so much.
For sure.
Thanks for you. Thanks for both of you.
Yeah, sure, no problem. All right, thank you so much all. Thank you for joining.
Oh, any problem in the transmission? Anyone is hearing me?
Yes, I can hear you.
I think Masume had paused down the internet. Anyone want to ask something?
Sorry, can I ask one question?
For sure.
Is there any exactive amount of days for cell coming from out of the freeze zone, freezer, something like into the room degree, 25 centigrade. I mean, is there any way that, I mean, is there any exact time for keeping cell in the freezer or in the refrigerator degree or in the room temperature? For example, the cell can survive like 2 days in the freezer, 3 days in refrigerator. I have some question like that.
In my experience, we had a lot of success using the Mr. Frost. Do you know Mr. Frost? The container that are automatic. Sorry. When we put the vials with the cells in there, we can put it for 30 minutes in the refrigerator, about -8 degrees. And then these 30 minutes, we pass to the -80 degrees Celsius, because this Mr. Frost, the container, it had a solution inside that transmits the 1 degree per hour. Okay. Okay, I will write my email. I had forget to.
My name is Hamoun. Yes, I understand. Thank you.
Okay. I will put my email in the chat, okay?
Thank you.
Thank you.
Okay, thank you so much. Sorry, I
Okay.
got interrupted, so I just connected again. And
Okay.
anyone else, if I have any question, please ask. Otherwise, we could say bye.
Okay. Goodbye.
Thank you
And
so
thank
much.
you all. Thank you all. Okay.
Thank you.
Goodbye.
Bye-bye. Bye. Bye. Thank you all, and bye for now.